This method provides a new approach to addressing key questions in the field of pulmonary vascular research, such as the molecular pathomechanisms of intimal proliferation in pulmonary hypertension. The main advantage of this method is that it provides a minute cellular biopsy of the pulmonary vascular endothelium without additional risk to the patient. Demonstrating the procedure will be Ms. Reina Perez, our clinical coordinator, and Dr. Phillip Gallo, the manager of my laboratory.
After the catheter is withdrawn from the sheath, immediately immerse it in a 50 milliliter centrifuge tube containing 40 milliliters of chilled, sterile saline. Swirl the catheter several times to remove any blood, and flush the line if necessary to remove the blood retained within it. Cut off the distal 5 centimeters of the catheter tip and allow it to fall into the centrifuge tube. Use forceps to immediately remove the tip and transfer it to a 15 milliliter centrifuge tube containing 15 milliliters of endothelial cell growth medium. Next, seal the tube. Place the tube on ice and transfer it to the lab. Either process the sample immediately or pack the sample on wet ice for overnight shipping to the laboratory where processing will occur.
It is critical that the balloon stays immersed in the medium. So make sure there is minimal head space in the 15 ml tube by filling it completely with endothelial cell growth medium.
First, add 5 milliliters of cell detachment solution to a clean 15 milliliter centrifuge tube. Place this tube on ice.